All right, we're recording. Oh my god, you can see the creeper. Oh well. Hello my friends, welcome to the first video of a series I'm hoping to do that helps you learn the content of your STEM courses. This is technically season 2, but whatever. Here I plan on covering introductory biology and organic chemistry, but there might be other topics added to this channel later on as I take more classes. In regards to the content I'm covering, I am using both my notes from when I took BioSci 1510 with Friedman and Clements, and the lecture videos from the Graham and Patton section, which, as most of you know, I'm a learning assistant for. A little background on me: I'm a sophomore at Vanderbilt majoring in molecular and cellular biology and medicine, health, and society. I also have a cinema and media studies minor because I'm not about to spend 4 years at Vanderbilt doing straight STEM. I don't hate myself. Alright, that's enough about me. Let's just get straight to the content. I personally think it would be really boring and disengaging to just stare at me lecturing, so you're about to witness my animation skills. It looks so bad! Oh my god, it looks so bad. Okay, bear with me. I just think it's easier to form connections between lecture topics when pictures are involved. Alright, so before we talked about membrane proteins, which is today's topic, we discussed the lipid bilayer that they are embedded in. If you still feel iffy on some of the characteristics of the lipid bilayer, I highly encourage you to go back and relearn it. In biology, we build on concepts, so make sure that foundation is strong. Now let's get into this. There are 3 kinds of membrane proteins we will discuss: integral, peripheral, and lipid anchored. As you will see, this lecture is pretty much all about integral membrane proteins. So integral membrane proteins are proteins embedded in the lipid bilayer. Sometimes these embedded proteins pass all the way through the lipid bilayer, thus further categorizing them as transmembrane proteins. You can see in this diagram that one protein can have multiple transmembrane segments, and these in this diagram are all amphipathic alpha helices. Though many transmembrane proteins function as gateways to permit the transport of specific substances across the membrane, there are also some transmembrane proteins, like, for example, lectins, that bind to specific oligosaccharide side chains that help form the sugar coating layer of the plasma membrane, called the glycocalyx, which actually I have no idea if I just pronounced that right, but that protects the cell from mechanical damage. So all in all, those integral transmembrane proteins are very versatile in function. Got a bit off track there, but I think this is very cool. So we have so much to cover in regards to integral membrane proteins because they play a massive role in transportation of molecules into the cell. So let's speed run this. Integral protein number 1, aquaporins. They allow for the passive movement of water across the membrane. Okay, a pair of positively charged residues, N2O3 and N68, attract the oxygen in the water molecule, which prevents hydrogen bonding. Side note about aquaporins, these are usually beta barrels. Integral protein number 2, facilitated transporters. These are how glucose enters the cell. Many molecules are too big to diffuse through the bilayer, so these facilitated transporters allow for a solute, once it is bound to the outside of the transporter, to trigger a conformational change in the protein, allowing it to open and for the solute to diffuse down its concentration gradient through that protein. Integral protein number 3, ion channels and pumps. Okay, so there are mechanogated, voltage-gated, and ligand-gated ion channels. Those are all part of the passive transport of ions, which involves movement of ions down the concentration gradient. Then there is the other category, which is active transport, which involves the movement of ions against their concentration gradient. Now, because these ions are moving against their concentration gradient, it requires the use of ATP, light, or electrons for energy. 2 pumps that you have to know that are active transport are the sodium-potassium pump and the calcium pump. As you already know, the sodium-potassium pump is essential for action potential propagation and uses 1 ATP to pump 3 sodium out of the cell and 2 potassium into the cell. Side note, you really should know what the Hodgkin and Keyes experiment is and the Post and Jolly experiments and how they contributed to our knowledge of the sodium-potassium pump, but I won't go into that here because I'm just trying to explain specifically cell stuff. Yeah. Okay. Now on to the next type of active transport ion pump. The calcium pump uses 1 ATP to pump 2 calcium ions out of the cell against its concentration gradient to make sure the cytosolic concentration of calcium is low. Moving on to the fourth integral protein, symporters and antiporters. Symporters and antiporters, they couple ion transport with other energetically favorable molecules so that the ion can be brought into or out of the cell. Symporters move cargo in the same direction, both in or both out, and antiporters, as you can probably assume, move cargo in the opposite direction. So those are all your integral proteins that we have required you to memorize. The notes don't give much detail about peripheral or lipid anchored proteins, so here's my short explanation of the two. Peripheral proteins are found on the cell surface and are not tightly attached to the cell membrane. They can be easily removed, which allows them to be involved in cell signaling. Lipid anchored proteins are literally like exactly what the name suggests. Proteins anchored in the cell membrane via lipids. They also play a role in cell signaling, and actually some sources show that certain lipid anchored proteins are actually integral membrane proteins, but for the purposes of what you guys have to know, I think we'll just consider them completely different categories for now. That concludes the first part out of 9 for the videos I'm making in relation to the second part of biology, second section of biology. Best of luck studying, and have a fantastic day. Alright, we're always going to have an ending credit disclaimer for these videos, just because I feel like I have to stress this. Number 1, I'm a student, not a professor. Number 2, my biology knowledge is by no means perfect. Number 3, I'm making this series using my college notes. If you find an error, please fact check me, correct me. I will not be hurt. I definitely do not want to be spreading wrong information, but this is just to my knowledge, to my understanding, trying to help you guys understand these concepts. So yeah, see you next time. I just rewatched that video and I apologize for the lack of eye contact that was there. I have a dual monitor, and like the monitor that shows my camera is the one that I'm facing right now, so I will fix that for the future. I apologize.